Hello, my name is Evan R. Major, and this is August 23rd, 2020. I'm going to be talking about Unit 2 Case Connections 1 and 2, and I'll start with Case 1, Part 1, which is the regulation of pyruvate kinase by healthy cells. Pyruvate kinase is an enzyme responsible for catalyzing the transfer of phosphate group from phosphoenolpyruvate to adenosine diphosphate, yielding one molecule of pyruvate and one molecule of ATP. In the Unit 2 case connection, we are given four distinct classes of the PK isoenzyme, including L for liver, R for erythrocytes, M1 for muscle and brain, and M2 for early fetal tissue and embryonic cells. The mechanism for regulation differs between the R and L pairs, as well as the M1 and the M2 pairs. R and L is regulated allosterically at a location other than the active site of the enzyme, and the M1-M2 pair is regulated by covalent modification, either phosphorylation or acetylation. Regarding the kinetics of this enzyme, the R and L forms have distinct conformation states, which are states of steric strain and bond energy. The two states contrast with each other in terms of the level of affinity for the substrate. The R state, or relaxed state, is characterized by high substrate affinity and a low Km. This is the activated form of PK stabilized by phosphoenolpyruvate and fructose 1,6-bisphosphate, which both promote the glycolytic pathway. The T state, or tense state, has a low substrate affinity and a high Km, serving as the inactivated form of PK and is bound by alanine and ATP. In the T state, PK is phosphorylated and inactivated, and this inhibits glycolysis. The domain shape, tetramer or dimer, which means 2 to 4 similar molecular units, can also affect glycolysis. PK tetramers have a much higher affinity for phosphoenolpyruvate and readily undergo dephosphorylation and activation. The pyruvate kinase dimers have a very low affinity and undergo phosphorylation, which in turn inhibits glycolysis. Finally, hormones such as epinephrine from the adrenal gland, glucagon and insulin from the pancreas can alter blood glucose levels and cause a change in PK activity. According to Birnbaum and his fellow authors, glucagon and epinephrine activate protein kinase A, which in turn phosphorylates and deactivates pyruvate kinase, shutting down glycolysis and activating gluconeogenesis. Contrastingly, pancreatic insulin activates phosphoprotein 1, leading to dephosphorylation and activation of pyruvate kinase to increase glycolysis. To review, PK inhibition or activation depend on conformation state, either the R or T, allosteric effectors, and inhibitors, which are non-competitive, as well as covalent modification, phosphorylation, and acetylation. Both of those are covalent, and hormone levels. Moving on to part 2 of case 1, I'm going to talk about PKM2 isoform in cancer cells and how it behaves in an unusual manner with respect to glycolysis. According to Mingchuan Su and Wenchun Hong, who are both authors of a 2018 study on PKM2, PKM2 transcription is attenuated in a number of adult tissues but reactivated in tumors. A study of the relative abundance of PKM1 and PKM2 in normal and tumor tissues demonstrated a switch from the PKM1 isoform to PKM2 in various cancers such as hepatocellular carcinoma and others. Finally, the change of mRNA splicing from PKM1 to PKM2 is enhanced by the CMYC oncogenes, suggesting that cancer cells engage the switch to suit their metabolism and proliferation. Another interesting fact about cancer cell behavior with respect to glycolysis, according to German researcher Erich Eigenroth, is that the PKM2 isozyme in cancer cells can be inhibited by Src-like tyrosine kinases. This unusual behavior is known because it inhibits the glycolytic step of pyruvate oxidation and diverts glucose instead to the pentose phosphate pathway. This is necessary to support nucleotide biosynthesis required by a rapidly dividing cell. To summarize the foundations of scientific dogma about PKM2 with respect to cancer, glycolytic inhibition and cell division largely rest on cell communication, signal transduction, to promote the expression of oncogenes and cell division, as well as the presence of tyrosine kinases that inhibit the further catabolism of glucose and divert it to the pentose phosphate pathway. I want to now move on to case 2 and discuss first part 1, which is glucose uptake in healthy tissues. Now we're going to discuss 4 different tissues, including liver, skeletal muscle, brain, and adipose tissue. First, with the liver, glucose is stored as glycogen, which functions as an energy reserve for muscle activity and for periods between meals and periods of stress. Pancreatic glucagon and adrenal epinephrine signal the liver to break down glycogen stores to support gluconeogenesis and supply energy to muscle cells. Insulin, however, signals the liver to take up excess blood glucose and store it as glycogen, or the process of glycogenesis. Skeletal muscle plays an important role in determining glycemia or blood sugar levels, and this is somewhat complex. It involves a chemical gradient using calcium 2+ ions and glucose transporters such as GLUT4 to transfer glucose and lactate in and out of muscle cells. In periods of intense exercise and oxygen depletion, the Cori cycle shuffles lactate from the muscle to the liver, where it is converted to pyruvate and eventually glucose. Liver glucose will either supply the muscle with glucose or store it in the form of glycogen when the muscles are at rest. Brain tissue is heavily dependent on glucose among other nutrients such as ketones and fatty acids. All types of brain cells, including neurons, glial cells, astrocytes, and others, regulate glucose metabolism completely independently. They have specific glucose transporters for this reason. Another important part of brain glucose uptake regulation is the gut-brain axis, and this is important because neuropeptides released by brain cells, they regulate food intake and gastrointestinal hormones, among hormones such as ghrelin and cholecystokinin, which in turn regulate digestion of metabolic fuels such as glucose in the brain. Adipose tissue is largely tasked with incorporating glucose carbons and producing carbon dioxide and glycerides. Adipose cell size and carbohydrate consumption are both factors in the regulation of glucose uptake and metabolism. With larger cell size and greater levels of carbohydrates in the diet, glucose to fatty acid catabolism and glucose oxidation seem to both increase and balance each other out. Problems arise when either the adipose cell is small and carbohydrate consumption is too high, or vice versa. Now, compared to these four healthy tissues, cancer tissues, with respect to glucose uptake, are not inhibited by body organs or homeostasis, among other factors. Whether it be the liver, pancreas, and muscle, both have an inhibiting and activating effects on liver. Liver has an inhibiting effect on muscle. Brain, the GI tract regulates glucose uptake in the brain. And with adipose tissue, cell size and carbohydrate metabolism have an effect on adipose tissue. Now, with respect to the glioblastoma, in case 2, what is noted here is this cancer not only upregulates glucose uptake, but it also diverts it to pentose phosphate pathway cell division, as well as deprives local tissue of nutrients such as magnesium and glucose, and in some cases grows in the area of the pituitary gland and inhibits the release of growth hormones. By doing this, this invasive and rapidly growing cancer contributes to necrosis in the surrounding tissue. Thank you.